Okay, let's talk a little bit about the B cells, which are part of the humoral immunity. So B cells are the ones that produce cells, that are plasma cells that produce antibodies. So they're part of the humoral part of the immunity, the other being the cell-mediated immunity. So in this case, the B cells carry receptors on the surface of the cell, which are called BCRs or B cell receptors. These receptors are basically just the antibodies. The only difference between these receptors and antibodies is that these are attached, these are bound to the surface of the cells in the plasma membrane, and antibodies are the ones which are free. So this version of antibodies is very similar in that regard. Um, and this is the structure of antibodies, usually shown as a letter Y, which consist of 4 polypeptides, 2 heavy chains and 2 light chains based on their sizes. 2 light chains are attached to one part of the heavy chains. And each antibody has 2 regions called variable region and constant region. Variable region is where the antigens are able to be bound. Those are called antigen binding sites, and each antibody has 2 of those sites. Now, since these are attached to the surface, they are embedded in the surface, so they are not free to move. They will move only when the cells move, when the B cells move. And variable regions have a huge capacity of what they can recognize. This is based on the, the structure of the variable regions, based on the amino acid sequence in that region that makes them very variable. So antibodies are capable of recognizing and binding to millions and millions of different types of antigens. The whole process of B cell activation takes place when B cells they recognize or they bind with any antigens or epitopes. They internalize them. Once anything is bound, is recognized by the BCR, then those are internalized. And based on those are naive cells at that time that were maturing in the in bone marrow. So once they bind to antigen and process them, they display those the kind of fragments of those proteins that they collected from the antigen along with MHC proteins. They display them on the surface, and then they interact with helper T cells. Helper T cells, if they are also able to recognize those antigens, then they activate naive B cells. They produce cytokines, and those cytokines that are produced by helper T cells activate these B cells. These B cells proliferate, and their proliferation results in the production of the plasma cells as well as memory cells. Plasma cells are those cells that will produce antibodies after they're formed from B cells. And this is the whole process shown here in this figure. It shows the attachment of the antigen, and then after that, internalization of the antigen being processed inside the endosomes, and from there the degradation of the antigen takes place, and those antigens are displayed on the surface of B cells along with MHC proteins, and then interaction with helper T cell takes place. Helper T cells, which is shown here, helper T cells, after they recognize the antigen, then they produce cytokines. And those B cells, they are activated. Um, Once they are activated, then the B cell will produce plasma cells, and memory cells will be produced from there, and then the whole process of proliferation, and this is called clonal selection, so that process will take place, and large number of those plasma cells will be produced that will be utilized to to produce antibodies, and antibodies are used against the antigens. If um, if they if the antigen is produced, if the antigen presented by B cell is presented to helper T cells, which are harmless, like internal proteins or something like that, then TH cells do not respond to that, and B cells become energetic, meaning, this is the part of the, the part of the uh, peripheral tolerance. They are able to recognize self cells versus the foreign cells. So antibodies are immunoglobulins, which are displayed usually with Y-shaped structures, and these are the monomers. Uh, Antibodies, which are free floating structures, they are not attached to anything. They are moving about by themselves. They're produced by plasma cells, and then after that, they are moving freely. They consist of two regions. Fab region, which is for antigen binding region. This is where the variable region is located, on two on each antibody, just like PCRs. And then they also have the second part called FC region. FC region is for crystallization. So these were designations given to them when they were identified initially. Immunoglobulins, there are several different types of immunoglobulins. There are IgMs, IgGs, IgA, IgD, and IgEs. These are all different types of antibodies that the cells are capable of producing. Where they vary from each other is in their constant region of the heavy chains. So since light chains do not have the constant regions, they are all variable regions, or part of that is a variable region. So that's why they're a little bit smaller. And the characteristics of these antibodies, without really going into too much detail, so the first antibodies that are produced are IgMs, and after that others are produced. IgGs are the most abundant antibodies that are produced, and usually and they are the most abundant ones found in blood and tissue fluids, and then there are other forms too. So IgMs are pentamers. There are five of those monomers attached as a single unit. IgMs are singles. They are monomers. IgAs are also not singles. They are a dimer. And then IgD and IgE are different ones. IgEs are the ones which are involved in allergic reactions. So, if the number of these antibodies increases, then that usually is equated with some type of allergic reaction. So sometimes doctors try to actually bring the level of IgE molecules down. And other ones are all present in different parts of the body. For example, IgAs are the ones which are secreted mostly in secretions, like in saliva and in milk and other types of mucus and tears. All of those have the form of antibodies which are IgAs. So we are very well protected with the help of these different types of antibodies. Primary response, which is when the first encounter of the B cells happens with the antigens. The cells at that time are naive cells, naive B cells. They recognize a particular antigen, and after that they multiply to produce a number of the clones of a particular type. So again, the first type of antibodies that are secreted from those plasma cells that come from B cells are the IgM molecules. IgM molecules, they are produced so that they can take care of the infection. They can bind to the infection, and that way they can start to kind of neutralize the effect of those antigens. So after the initial antibodies are produced, IgMs, then a couple of things happen after that, and those are mentioned at the bottom of the slide. Affinity maturation and class switching. So affinity maturation means, after they encounter, after B cells are activated and they start to go through the clonal proliferation process or selection process, then they become stronger and stronger. So they are selected for responding strongly against any antigen. That is called affinity maturation. So as they encounter the threat or the antigen, they become stronger and stronger, and only those are selected that respond very strongly against antigen. The second thing is class switching happens. Class switching means that only other than IgM, which are produced initially, other classes of antibodies are produced. So this is the process of affinity maturation. So where the activated B cells, when they proliferate, when they make more copies of themselves, they are selected which one of these two is reacting strongly against an antigen. Obviously, this is the one which is selected, and then it continues to proliferate to make more cells. So this one will not be allowed to continue afterwards. So the one that gave a stronger response will be selected, and once it proliferates, it divides to make more cells, and then the same selection process will take place. This will be eliminated, this will be selected, and it will produce more cells. So now with this screening process, only the stronger ones are selected and other ones are not allowed to continue to proliferate afterwards. Class switching results in the production of other types of other classes of antibodies. After IgMs, then other ones are produced, for example, in case of lymph nodes. Because of the changes in the DNA of those cells, they produce IgGs. Or in case of mouth or other parts of the body, like in different secretions, in mammary glands or in saliva, others IgA types of antibodies are produced. Okay, so these two things, class switching and affinity maturation, are part of the primary responses. Primary response happens when when the antigen is encountered by naive B cells and they get activated, and the rest of the process starts from there. At the end of this, the plasma cells are produced that produce antibodies, antibodies clear antigens, and everything is okay afterwards. Then eventually the level of the number of these cells also drops. So few, and the result of that is the memory cells are the ones that are left behind at the end. So this is how the secondary response starts too. So now after the first encounter had happened, that produced those cells, plasma cells that produce antibodies, and memory cells that were left behind after the antigen was completely cleared from the system. Whenever the same antigen epitope is recognized by the memory cells, then the response this time is much more severe. The first time when that infection happened or attack happened, then IgGs were produced and memory cells were produced. So which is shown here in the first part of this graph here. The antigen was introduced right here. Some response was made, IgMs were produced, then IgG molecules were produced, and then things got clear. It took some period of time to produce those, and sometime that period could be days and months to have this type of a response. When the second time the same thing happens, the level of IgM actually was dropped back to almost nothing, but IgG stayed on. The level of IgG was much higher than the first time. First time they were not even existing inside the cells. So the second time they were there. The level of those antibodies was much higher than the first time. So the secondary response produced those antibodies very quickly. memory cells were there already. So IgG molecules level increase significantly in a matter of a couple of days. And then IgM level also increased quite significantly within days, not months, but very shorter period of time. So this is the kind of things that we see in vaccination as well. In vaccination, the response that comes from the second infection uh, is, is because of the vaccination that was done, that prepared our system, produced all the cells that were needed and antibodies that were needed to be ready, um, had the same type of infection occurring the second time. So the characteristic of the secondary response is that memory cells that were produced against a very specific antigen were present in great number this time. Affinity maturation had prepared some of those antibodies that were needed to counter the attack the second time. IgAs and IgG molecules were produced because of class switching, and then we had enough amount of material and weapons available to us to be ready for the next time.